Hippocampus.
The hippocampus is a major component of the brains of humans and other vertebrates. It belongs to the limbic system and plays important roles in the consolidation of information from short-term memory to long-term memory and spatial navigation. Humans and other mammals have two hippocampi, one on each side of the brain. The hippocampus is located under the cerebral cortex. And in primates it is located in the medial temporal lobe, underneath the cortical surface. It contains two main interlocking parts, Ammon's horn and the dentate gyrus. In Alzheimer's disease, the hippocampus is one of the first regions of the brain to suffer damage. Memory loss and disorientation are included among the early symptoms. Damage to the hippocampus can also result from oxygen starvation, hypoxia, encephalitis, or medial temporal lobe epilepsy. People with extensive, bilateral hippocampal damage may experience anterograde amnesia, the inability to form or retain new memories. In rodents, the hippocampus has been studied extensively as part of a brain system responsible for spatial memory and navigation. Many neurons in the rat and mouse hippocampus respond as place cells, that is, they fire bursts of action potentials when the animal passes through a specific part of its environment. Hippocampal place cells interact extensively with head direction cells, whose activity acts as an inertial compass, and conjecturally with grid cells in the neighboring entorhinal cortex. Since different neuronal cell types are neatly organized into layers in the hippocampus, it has frequently been used as a model system for studying neurophysiology. The form of neural plasticity known as long-term potentiation, LTP, was first discovered to occur in the hippocampus and has often been studied in this structure. LTP is widely believed to be one of the main neural mechanisms by which memory is stored in the brain.
Name.
The earliest description of the ridge running along the floor of the temporal horn of the lateral ventricle comes from the Venetian anatomist Julius Caesar Arenzi, 1587, who likened it first to a silkworm and then to a seahorse, Latin, hippocampus from Greek, hippos, horse, and campos, sea monster. The German anatomist Duveroy, 1729, the first to illustrate the structure, also wavered between seahorse and silkworm. Ram's horn was proposed by Danish anatomist Jacob Winslow in 1732. And a decade later his fellow Parisian, the surgeon Dagarin Jot, used cornu ammonis horn of, the ancient Egyptian god, Ammon. Another mythological reference appeared with the term pes hippocampi, which may date back to Dimebroek in 1672, introducing a comparison with the shape of the folded back forelimbs and webbed feet of the classical hippocampus, Greek, Iocampos, a sea monster with a horse's forequarters and a fish's tail. The hippocampus was then described as pes hippocampi major, with an adjacent bulge in the occipital horn, the calcar avis, being named pes hippocampi minor. The renaming of the hippocampus as hippocampus major, and the calcar avis as hippocampus minor, has been attributed to Felix Victor's systematizing nomenclature of parts of the brain in 1786. Mayer mistakenly used the term hippopotamus in 1779, and was followed by some other authors until Carl Friedrich Bödach resolved this error in 1829. In 1861 the hippocampus minor became the center of a dispute over human evolution between Thomas Henry Huxley and Richard Owen, satirized as the Great Hippocampus Question. The term hippocampus minor fell from use in anatomy textbooks, and was officially removed in the Nomina Anatomica of 1895. Today, the structure is called the hippocampus rather than hippocampus major, with pes hippocampi often being regarded as synonymous with Dagarin Jot's cornu ammonis, a term that survives in the names of the four main histological divisions of the hippocampus, CA1, CA2, CA3, and CA4.
Functions.
Historically, the earliest widely held hypothesis was that the hippocampus is involved in olfaction. This idea was cast into doubt by a series of anatomical studies that did not find any direct projections to the hippocampus from the olfactory bulb. However, later work did confirm that the olfactory bulb does project into the ventral part of the lateral entorhinal cortex, and field CA1 in the ventral hippocampus sends axons to the main olfactory bulb, the anterior olfactory nucleus, and to the primary olfactory cortex. There continues to be some interest in hippocampal olfactory responses, in particular the role of the hippocampus in memory for odors, but few specialists today believe that olfaction is its primary function. Over the years, three main ideas of hippocampal function have dominated the literature: inhibition, memory, and space. The behavioral inhibition theory, caricatured by O'Keefe and Nadal as slam on the brakes, was very popular up to the 1960s. It derived much of its justification from two observations: first, that animals with hippocampal damage tend to be hyperactive. Second, that animals with hippocampal damage often have difficulty learning to inhibit responses that they have previously been taught, especially if the response requires remaining quiet, as in a passive avoidance test. Jeffrey Gray developed this line of thought into a full-fledged theory of the role of the hippocampus in anxiety. The inhibition theory is currently the least popular of the three. The second major line of thought relates the hippocampus to memory. Although it had historical precursors, this idea derived its main impetus from a famous report by William Beecher Scoville and Brenda Milner describing the results of surgical destruction of the hippocampi, in an attempt to relieve epileptic seizures, in Henry Molaison, known until his death in 2008 as patient HM. The unexpected outcome of the surgery was severe anterograde and partial retrograde amnesia. Molaison was unable to form new episodic memories after his surgery and could not remember any events that occurred just before his surgery, but he did retain memories of events that occurred many years earlier, extending back into his childhood. This case attracted such widespread professional interest that Molaison became the most intensively studied subject in medical history. In the ensuing years, other patients with similar levels of hippocampal damage and amnesia, caused by accident or disease, have also been studied, and thousands of experiments have studied the physiology of activity-driven changes in synaptic connections in the hippocampus. There is now almost universal agreement that the hippocampi plays some sort of important role in memory. However, the precise nature of this role remains widely debated. The third important theory of hippocampal function relates the hippocampus to space. The spatial theory was originally championed by O'Keefe and Nadal, who were influenced by E. C. Tolman's theories about cognitive maps in humans and animals. O'Keefe and his student Ostrovsky in 1971 discovered neurons in the rat hippocampus that appeared to them to show activity related to the rat's location within its environment. Despite skepticism from other investigators, O'Keefe and his co-workers, especially Lynn Nadal, continued to investigate this question, in a line of work that eventually led to their very influential 1978 book The Hippocampus as a Cognitive Map. As with the memory theory, there is now almost universal agreement that spatial coding plays an important role in hippocampal function, but the details are widely debated.
Role in memory.
Psychologists and neuroscientists generally agree that the hippocampus plays an important role in the formation of new memories about experienced events, episodic or autobiographical memory. Part of this function is hippocampal involvement in the detection of novel events, places and stimuli. Some researchers regard the hippocampus as part of a larger medial temporal lobe memory system responsible for general declarative memory, memories that can be explicitly verbalized. These would include, for example, memory for facts in addition to episodic memory. Due to bilateral symmetry, the brain has a hippocampus in each cerebral hemisphere, so every normal brain has two of them. If damage to the hippocampus occurs in only one hemisphere, leaving the structure intact in the other hemisphere, the brain can retain the normal memory functioning. Severe damage to the hippocampi in both hemispheres results in profound difficulties in forming new memories, anterograde amnesia, and often also affects memories formed before the damage occurred, retrograde amnesia. Although the retrograde effect normally extends many years back before the brain damage, in some cases older memories remain. This retention of older memories leads to the idea that consolidation over time involves the transfer of memories out of the hippocampus to other parts of the brain. damage to the hippocampus does not affect some types of memory, such as the ability to learn new skills, playing a musical instrument or solving certain types of puzzles, for example. This fact suggests that such abilities depend on different types of memory, procedural memory, and different brain regions. Furthermore, amnesic patients frequently show implicit memory for experiences even in the absence of conscious knowledge. For example, patients asked to guess which of two faces they have seen most recently may give the correct answer most of the time in spite of stating that they have never seen either of the faces before. Some researchers distinguish between conscious recollection, which depends on the hippocampus, and familiarity, which depends on portions of the medial temporal cortex.
Role in spatial memory and navigation.
Studies conducted on freely moving rats and mice have shown that many hippocampal neurons have place fields, that is, they fire bursts of action potentials when a rat passes through a particular part of the environment. Evidence for place cells in primates is limited, perhaps in part because it is difficult to record brain activity from freely moving monkeys. Place-related hippocampal neural activity has been reported in monkeys moving around inside a room while seated in a restraint chair. On the other hand, Edmund Rolls and his colleagues instead described hippocampal cells that fire in relation to the place a monkey is looking at, rather than the place where its body is located. In humans, cells with location-specific firing patterns have been reported in a study of patients with drug-resistant epilepsy who were undergoing an invasive procedure to localize the source of their seizures, with a view to surgical resection. The patients had diagnostic electrodes implanted in their hippocampus and then used a computer to move around in a virtual reality town. Place responses in rats and mice have been studied in hundreds of experiments over 4 decades, yielding a large quantity of information. Place cell responses are shown by pyramidal cells in the hippocampus proper, and granule cells in the dentate gyrus. These constitute the great majority of neurons in the densely packed hippocampal layers. Inhibitory interneurons, which make up most of the remaining cell population, frequently show significant place-related variations in firing rate that are much weaker than those displayed by pyramidal or granule cells. There is little if any spatial topography in the representation. In general, cells lying next to each other in the hippocampus have uncorrelated spatial firing patterns. Place cells are typically almost silent when a rat is moving around outside the place field but reach sustained rates as high as 40 Hz when the rat is near the center. Neural activity sampled from 30 to 40 randomly chosen place cells carries enough information to allow a rat's location to be reconstructed with high confidence. The size of place fields varies in a gradient along the length of the hippocampus, with cells at the dorsal end showing the smallest fields, cells near the center showing larger fields, and cells at the ventral tip fields that cover the entire environment. In some cases, the firing rate of rat hippocampal cells depends not only on place but also on the direction a rat is moving, the destination toward which it is traveling, or other task-related variables. The discovery of place cells in the 1970s led to a theory that the hippocampus might act as a cognitive map, a neural representation of the layout of the environment. Several lines of evidence support the hypothesis. It is a frequent observation that without a fully functional hippocampus, humans may not remember where they have been and how to get where they are going. Getting lost is one of the most common symptoms of amnesia. Studies with animals have shown that an intact hippocampus is required for initial learning and long-term retention of some spatial memory tasks, in particular ones that require finding the way to a hidden goal. The cognitive map hypothesis has been further advanced by recent discoveries of head direction cells, grid cells, and border cells in several parts of the rodent brain that are strongly connected to the hippocampus. Brain imaging shows that people have more active hippocampi when correctly navigating, as tested in a computer-simulated virtual navigation task. Also, there is evidence that the hippocampus plays a role in finding shortcuts on new routes between familiar places. For example, London's taxi drivers must learn a large number of places and the most direct routes between them. They have to pass a strict test, the knowledge, before being licensed to drive the famous black cabs. A study at University College London by Maguire, al. 2000, showed that part of the hippocampus is larger in taxi drivers than in the general public, and that more experienced drivers have bigger hippocampi. Whether having a bigger hippocampus helps an individual to become a better cab driver, or if finding shortcuts for a living makes an individual's hippocampus grow is yet to be elucidated. However, in that study, Maguire al. examined the correlation between size of the grey matter and length of time that had been spent as a taxi driver, and found a positive correlation between the length of time an individual had spent as a taxi driver and the volume of the right hippocampus. It was found that the total volume of the hippocampus remained constant, from the control group versus taxi drivers. That is to say that the posterior portion of a taxi driver's hippocampus is indeed increased, but at the expense of the anterior portion. There have been no known detrimental effects reported from this disparity in hippocampal proportions.
Anatomy.
In terms of anatomy, the hippocampus is an elaboration of the edge of the cerebral cortex. The structures that line the edge of the cortex make up the so-called limbic system, Latin limbus equals border. These include the hippocampus, cingulate cortex, olfactory cortex, and amygdala. Paul MacLean once suggested, as part of his triune brain theory, that the limbic structures comprise the neural basis of emotion. Some neuroscientists no longer believe that the concept of a unified limbic system is valid, however. Yet, the hippocampus is anatomically connected to parts of the brain that are involved with emotional behavior, the septum, the hypothalamic mammillary body, and the anterior nuclear complex in the thalamus, therefore its role as a limbic structure cannot be completely dismissed. The hippocampus as a whole has the shape of a curved tube, which has been variously compared to a seahorse, a ram's horn, cornu ammonis, hence the subdivisions CA1 through CA4, or a banana. It can be distinguished as a zone where the cortex narrows into a single layer of densely packed pyramidal neurons 3 to 6 cells deep in rats, which curl into a tight U-shape. One edge of the U, field CA4, is embedded into a backward-facing, strongly flexed, V-shaped cortex, the dentate gyrus. It consists of ventral and dorsal portions, both of which are of similar composition but are parts of different neural circuits. This general layout holds across the full range of mammalian species, from hedgehog to human, although the details vary. In the rat, the two hippocampi resemble a pair of bananas, joined at the stems by the hippocampal commissure that crosses the midline under the anterior corpus callosum. In human or monkey brains, the portion of the hippocampus down at the bottom, near the base of the temporal lobe, is much broader than the part at the top. One of the consequences of this complex geometry is that cross-sections through the hippocampus can show a variety of shapes, depending on the angle and location of the cut. The entorhinal cortex, EC, located in the parahippocampial gyrus, is considered to be part of the hippocampal region because of its anatomical connections. The EC is strongly and reciprocally connected with many other parts of the cerebral cortex. In addition, the medial septal nucleus, the anterior nuclear complex and nucleus reunions of the thalamus and the supramammillary nucleus of the hypothalamus, as well as the raphe nuclei and locus coeruleus in the brainstem send axons to the EC. The main output pathway, perforant path, first described by Ramon y Cajal, of EC axons comes from the large pyramidal cells in layer 2 that perforate the subiculum and project densely to the granule cells in the dentate gyrus. Apical dendrites of CA3 get a less dense projection, and the apical dendrites of CA1 get a sparse projection. Thus, the perforant path establishes the EC as the main interface between the hippocampus and other parts of the cerebral cortex. The dentate granule cell axons, called mossy fibers, pass on the information from the EC on thorny spines that exit from the proximal apical dendrite of CA3 pyramidal cells. Then, CA3 axons exit from the deep part of the cell body and loop up into the region where the apical dendrites are located, then extend all the way back into the deep layers of the entorhinal cortex, the Schaffer collaterals completing the reciprocal circuit. Field CA1 also sends axons back to the EC, but these are more sparse than the CA3 projection. Within the hippocampus, the flow of information from the EC is largely unidirectional, with signals propagating through a series of tightly packed cell layers, first to the dentate gyrus, then to the CA3 layer, then to the CA1 layer, then to the subiculum, then out of the hippocampus to the EC, mainly due to collateralization of the CA3 axons. Each of these layers also contains complex intrinsic circuitry and extensive longitudinal connections. Several other connections play important roles in hippocampal function. Beyond the output to the EC, additional output pathways go to other cortical areas including the frontal cortex. A very important large output goes to the lateral septal area and to the mammillary body of the hypothalamus. The hippocampus receives modulatory input from the serotonin, norepinephrine, and dopamine systems, and from nucleus reunions of the thalamus to field CA1. A very important projection comes from the medial septal area, which sends cholinergic and GABAergic fibers to all parts of the hippocampus. The inputs from the septal area play a key role in controlling the physiological state of the hippocampus. Destruction of the septal area abolishes the hippocampal theta rhythm and severely impairs certain types of memory. The cortical region adjacent to the hippocampus is known collectively as the parahippocampal gyrus, or parahippocampi. It includes the EC and also the perirhinal cortex, which derives its name from the fact that it lies next to the rhinal sulcus. The perirhinal cortex plays an important role in visual recognition of complex objects. There is also substantial evidence that it makes a contribution to memory, which can be distinguished from the contribution of the hippocampus. It is apparent that complete amnesia occurs only when both the hippocampus and the parahippocampus are damaged.
Hippocampal formation.
Various sections of the hippocampal formation are shown to be functionally and anatomically distinct. The dorsal, DH, ventral, VH, and intermediate regions of the hippocampal formation serve different functions, project with differing pathways, and have varying degrees of place field neurons, Fornzilo and Ong, 2009. The dorsal region of the hippocampal formation serves for spatial memory, verbal memory, and learning of conceptual information. Using the radial arm maze Perthwison al. 2004, found lesions in the DH to cause spatial memory impairment while VH lesions did not. Its projecting pathways include the medial septal complex and supramammillary nucleus. The dorsal hippocampal formation also has more place field neurons than both the ventral and intermediate hippocampal formations, Young al. 1994. The intermediate hippocampus has overlapping characteristics with both the ventral and dorsal hippocampus, Fornzilo and Ong, 2009. Using PHAL anterograde tracing methods, Sinquiza and Swanson, 2007, located the moderate projections to two primary olfactory cortical areas and prelimbic areas of the MPFC. This region has the smallest number of place field neurons. The ventral hippocampus functions in fear conditioning and affective processes. Anagnostaras al. 2002, showed that alterations to the ventral hippocampus reduce the amount of information sent to the amygdala by the dorsal and ventral hippocampus, consequentially altering fear conditioning in rats.
Physiology.
The hippocampus shows two major modes of activity, each associated with a distinct pattern of neural population activity and waves of electrical activity as measured by an electroencephalogram, EEG. These modes are named after the EEG patterns associated with them, theta and large irregular activity, LIA. The main characteristics described below are for the rat, which is the animal most extensively studied. The theta mode appears during states of active, alert behavior, especially locomotion, and also during REM, dreaming, sleep. In the theta mode, the EEG is dominated by large regular waves with a frequency range of 6 to 9 Hz, and the main groups of hippocampal neurons, pyramidal cells and granule cells, show sparse population activity, which means that in any short time interval, the great majority of cells are silent, while the small remaining fraction fire at relatively high rates, up to 50 spikes in 1 second for the most active of them. An active cell typically stays active for half a second to a few seconds. As the rat behaves, the active cells fall silent and new cells become active, but the overall percentage of active cells remains more or less constant. In many situations, cell activity is determined largely by the spatial location of the animal, but other behavioral variables also clearly influence it. The LIA mode appears during slow wave, non-dreaming, sleep, and also during states of waking immobility such as resting or eating. In the LIA mode, the EEG is dominated by sharp waves that are randomly timed large deflections of the EEG signal lasting for 25 to 50 milliseconds. Sharp waves are frequently generated in sets, with sets containing up to 5 or more individual sharp waves and lasting up to 500 milliseconds. The spiking activity of neurons within the hippocampus is highly correlated with sharp wave activity. Most neurons decrease their firing rate between sharp waves. However, during a sharp wave, there is a dramatic increase of firing rate in up to 10% of the hippocampal population. These two hippocampal activity modes can be seen in primates as well as rats, with the exception that it has been difficult to see robust theta rhythmicity in the primate hippocampus. There are, however, qualitatively similar sharp waves and similar state-dependent changes in neural population activity.
Theta rhythm.
Because of its densely packed neural layers, the hippocampus generates some of the largest EEG signals of any brain structure. In some situations the EEG is dominated by regular waves at 3 to 10 Hz, often continuing for many seconds. These reflect subthreshold membrane potentials and strongly modulate the spiking of hippocampal neurons and synchronize across the hippocampus in a traveling wave pattern. This EEG pattern is known as a theta rhythm. Theta rhythmicity is very obvious in rabbits and rodents and also clearly present in cats and dogs. Whether theta can be seen in primates is a vexing question. In rats, the animals that have been the most extensively studied, theta is seen mainly in two conditions: first, when an animal is walking or in some other way actively interacting with its surroundings. Second, during REM sleep. The function of theta has not yet been convincingly explained, although numerous theories have been proposed. The most popular hypothesis has been to relate it to learning and memory. An example would be the phase with which theta rhythms, at the time of stimulation of a neuron, shape the effect of that stimulation upon its synapses. What is meant here is that theta rhythms may affect those aspects of learning and memory that are dependent upon synaptic plasticity. It is well established that lesions of the medial septum, the central node of the theta system, cause severe disruptions of memory. However, the medial septum is more than just the controller of theta. It is also the main source of cholinergic projections to the hippocampus. It has not been established that septal lesions exert their effects specifically by eliminating the theta rhythm.
Sharp waves.
During sleep or during waking states when an animal is resting or otherwise not engaged with its surroundings, the hippocampal EEG shows a pattern of irregular slow waves, somewhat larger in amplitude than theta waves. This pattern is occasionally interrupted by large surges called sharp waves. These events are associated with bursts of spike activity lasting 50 to 100 milliseconds in pyramidal cells of CA3 and CA1. They are also associated with short-lived high-frequency EEG oscillations called ripples, with frequencies in the range 150 to 200 Hz in rats. Sharp waves are most frequent during sleep when they occur at an average rate of around 1 per second, in rats, but in a very irregular temporal pattern. Sharp waves are less frequent during an active waking states and are usually smaller. Sharp waves have also been observed in humans and monkeys. In macaques, sharp waves are robust but do not occur as frequently as in rats. One of the most interesting aspects of sharp waves is that they appear to be associated with memory. Wilson and McNaughton 1994, and numerous later studies, reported that when hippocampal place cells have overlapping spatial firing fields, and therefore often fire in near simultaneity, they tend to show correlated activity during sleep following the behavioral session. This enhancement of correlation, commonly known as reactivation, has been found to occur mainly during sharp waves. It has been proposed that sharp waves are, in fact, reactivations of neural activity patterns that were memorized during behavior, driven by strengthening of synaptic connections within the hippocampus. This idea forms a key component of the two-stage memory theory, advocated by Bwatsaki and others, which proposes that memories are stored within the hippocampus during behavior and then later transferred to the neocortex during sleep. Sharp waves are suggested to drive Hebbian synaptic changes in the neocortical targets of hippocampal output pathways.
Long-term potentiation.
Since at least the time of Ramon y Cajal, psychologists have speculated that the brain stores memory by altering the strength of connections between neurons that are simultaneously active. This idea was formalized by Donald Hebb in 1948, but for many years thereafter, attempts to find a brain mechanism for such changes failed. In 1973, Tim Bliss and Terje Lomo described a phenomenon in the rabbit hippocampus that appeared to meet Hebb's specifications, a change in synaptic responsiveness induced by brief strong activation and lasting for hours or days or longer. This phenomenon was soon referred to as long-term potentiation, abbreviated LTP. As a candidate mechanism for memory, LTP has since been studied intensively, and a great deal has been learned about it. The hippocampus is a particularly favorable site for studying LTP because of its densely packed and sharply defined layers of neurons, but similar types of activity-dependent synaptic change have now been observed in many other brain areas. The best studied form of LTP occurs at synapses that terminate on dendritic spines and use the transmitter glutamate. Several of the major pathways within the hippocampus fit this description and exhibit LTP. The synaptic changes depend on a special type of glutamate receptor, the NMDA receptor, which has the special property of allowing calcium to enter the postsynaptic spine only when presynaptic activation and postsynaptic depolarization occur at the same time. Drugs that interfere with NMDA receptors block LTP and have major effects on some types of memory, especially spatial memory. Transgenic mice, genetically modified in ways that disable the LTP mechanism, also generally show severe memory deficits.
Pathology. Aging.
Age-related conditions such as Alzheimer's disease, for which hippocampal disruption is one of the earliest signs, have a severe impact on many types of cognition, but even normal aging is associated with a gradual decline in some types of memory, including episodic memory and working memory, or short-term memory. Because the hippocampus is thought to play a central role in memory, there has been considerable interest in the possibility that age-related declines could be caused by hippocampal deterioration. Some early studies reported substantial loss of neurons in the hippocampus of elderly people, but later studies using more precise techniques found only minimal differences. Similarly, some MRI studies have reported shrinkage of the hippocampus in elderly people, but other studies have failed to reproduce this finding. There is, however, a reliable relationship between the size of the hippocampus and memory performance, meaning that not all elderly people show hippocampal shrinkage, but those who do tend to perform less well on some memory tasks. There are also reports that memory tasks tend to produce less hippocampal activation in elderly than in young subjects. Furthermore, a randomized control study published in 2011 found that aerobic exercise could increase the size of the hippocampus in adults aged 55 to 80 and also improve spatial memory. In rats, where detailed studies of cellular physiology are possible, aging does not cause substantial cell loss in the hippocampus, but it alters synaptic connectivity in several ways. Functional synapses are lost in the dentate gyrus and CA1 region, and NMDA receptor-mediated responses are reduced. These changes may account for deficits in induction and maintenance of long-term potentiation, a form of synaptic plasticity that has been implicated in memory. There are also age-related declines in hippocampal expression of several genes associated with synaptic plasticity, Finally, there are differences in the stability of place cell representations. In young rats, the arrangement of place fields is usually altered if the rat is moved into a different environment but remains the same if a rat was returned to an environment it has visited previously. In aged rats, the place fields frequently fail to remap when a rat is moved to a different environment and also frequently fail to restore the original map when the rat is returned to the same environment. Other studies in rats have shown a reversal in cognitive decline when they have been given a diet high in blueberries, which contain the antioxidant pterostilbene. It is thought that this could be due to the unique ability of pterostilbene to cross the blood-brain barrier and to co-localize in the hippocampus, which is seen as the brain's memory center.
Stress.
The hippocampus contains high levels of glucocorticoid receptors, which make it more vulnerable to long-term stress than most other brain areas. Stress-related steroids affect the hippocampus in at least 3 ways. First, by reducing the excitability of some hippocampal neurons. Second, by inhibiting the genesis of new neurons in the dentate gyrus. Third, by causing atrophy of dendrites and pyramidal cells of the CA3 region. There is evidence that humans having experienced severe, long-lasting traumatic stress show atrophy of the hippocampus more than of other parts of the brain. These effects show up in post-traumatic stress disorder, and they may contribute to the hippocampal atrophy reported in schizophrenia and severe depression. A recent study has also revealed atrophy as a result of depression, but this can be stopped with antidepressants even if they are not effective in relieving other symptoms. Hippocampal atrophy is also frequently seen in Cushing's syndrome, a disorder caused by high levels of cortisol in the bloodstream. At least some of these effects appear to be reversible if the stress is discontinued. There is, however, evidence derived mainly from studies using rats that stress occurring shortly after birth can affect hippocampal function in ways that persist throughout life. Sex-specific responses to stress have also been demonstrated to have an effect on the hippocampus. During situations in which adult male and female rats were exposed to chronic stress, the females were shown to be better able to cope.
Epilepsy.
The hippocampus is often the focus of epileptic seizures. Hippocampal sclerosis is the most commonly visible type of tissue damage in temporal lobe epilepsy. It is not yet clear, however, whether the epilepsy is usually caused by hippocampal abnormalities or whether the hippocampus is damaged by cumulative effects of seizures. In experimental settings where repetitive seizures are artificially induced in animals, hippocampal damage is a frequent result. This may be a consequence of the hippocampus as being one of the most electrically excitable parts of the brain. It may also have something to do with the fact that the hippocampus is one of very few brain regions where new neurons continue to be created throughout life.
Schizophrenia.
The causes of schizophrenia are not at all well understood, but numerous abnormalities of brain structure have been reported. The most thoroughly investigated alterations involve the cerebral cortex, but effects on the hippocampus have also been described. Many reports have found reductions in the size of the hippocampus in schizophrenic subjects. The changes probably result from altered development rather than tissue damage and show up even in subjects never having been medicated. Several lines of evidence implicate changes in synaptic organization and connectivity. It is unclear whether hippocampal alterations play any role in causing the psychotic symptoms that are the most important feature of schizophrenia. Anthony Grace and his co-workers have suggested, on the basis of experimental work using animals, that hippocampal dysfunction might produce an alteration of dopamine release in the basal ganglia, thereby indirectly affecting the integration of information in the frontal cortex. Others have suggested that hippocampal dysfunction might account for disturbances in long-term memory frequently observed in people with schizophrenia.
Transient global amnesia.
A current hypothesis as to one cause of transient global amnesia, a dramatic, sudden, temporary, near-total loss of short-term memory, is that it may be due to venous congestion of the brain, leading to ischemia of structures such as the hippocampus that are involved in memory.
Evolution.
The hippocampus has a generally similar appearance across the range of mammal species, from monotreme such as the echidna to primates such as humans. The higher campal size to body size ratio broadly increases, being about twice as large for primates as for the echidna. It does not, however, increase at anywhere close to the rate of the neocortex to body size ratio. Therefore, the hippocampus takes up a much larger fraction of the cortical mantle in rodents than in primates. In adult humans, the volume of the hippocampus on each side of the brain is about 3.0 to 3.5 cm 3 as compared to 320 to 420 cm 3 for the volume of the neocortex. There is also a general relationship between the size of the hippocampus and spatial memory. When comparisons are made between similar species, those that have a greater capacity for spatial memory tend to have larger higher campal volumes. This relationship also extends to sex differences. In species where males and females show strong differences in spatial memory ability, they also tend to show corresponding differences in higher campal volume. Non-mammalian species do not have a brain structure that looks like the mammalian hippocampus, but they have one that is considered homologous to it. The hippocampus, as pointed out above, is in essence the medial edge of the cortex. Only mammals have a fully developed cortex, but the structure it evolved from, called the pallium, is present in all vertebrates, even the most primitive ones such as the lamprey or hagfish. The pallium is usually divided into three zones: medial, lateral and dorsal. The medial pallium forms the precursor of the hippocampus. It does not resemble the hippocampus visually because the layers are not warped into an S shape or enfolded by the dentate gyrus, but the homology is indicated by strong chemical and functional affinities. There is now evidence that these higher campal-like structures are involved in spatial cognition in birds, reptiles, and fish. In birds, the correspondence is sufficiently well established that most anatomists refer to the medial pallial zone as the avian hippocampus. Numerous species of birds have strong spatial skills, in particular those that cache food. There is evidence that food-caching birds have a larger hippocampus than other types of birds and that damage to the hippocampus causes impairments in spatial memory. The story for fish is more complex. In teleost fish, which make up the great majority of existing species, the forebrain is distorted in comparison to other types of vertebrates. Most neuroanatomists believe that the teleost forebrain is in essence everted, like a sock turned inside out, so that structures that lie in the interior, next to the ventricles, for most vertebrates, are found on the outside in teleost fish, and vice versa. One of the consequences of this is that the medial pallium, higher campal zone, of a typical vertebrate is thought to correspond to the lateral pallium of a typical fish. Several types of fish, particularly goldfish, have been shown experimentally to have strong spatial memory abilities, even forming cognitive maps of the areas they inhabit. There is evidence that damage to the lateral pallium impairs spatial memory. Thus, the role of the higher campal region in navigation appears to begin far back in vertebrate evolution, predating splits that occurred hundreds of millions of years ago. It is not yet known whether the medial pallium plays a similar role in even more primitive vertebrates, such as sharks and rays, or even lampreys and hagfish. Some types of insects, and mollusks such as the octopus, also have strong spatial learning and navigation abilities, but these appear to work differently from the mammalian spatial system, so there is as yet no good reason to think that they have a common evolutionary origin. Nor is there sufficient similarity in brain structure to enable anything resembling a hippocampus to be identified in these species. Some have proposed, however, that the insects' mushroom bodies may have a function similar to that of the hippocampus.
Notes.
http://neuroscience.ut.tmc.edu/s4/ Chapter 05.html, Pierce, 2001, ABC Duveroy, 2005, Gross, 1993, Ichsler, 2004, Finger, pages 183, Site PMID 690266, Eichenborn al., 1991, Van der Wolf, 2001, Nadal al., 1975, Gray and McNaughton, 2000, Best and White, 1999, Scoville and Milner, 1957, NY Times, December 6, 2008, Squire, 2009, A.B. Squire, 1992, A.B.I. Chenbaum and Cohen, 1993, O'Keefe and Ostrovsky, 1971, O'Keefe and Nadal, 1978, ABCD Moser al., 2008, Squire and Schachter, 2002, Van Eltzacher al., 2008, Deganera G., Grimaldo L.G., Quarato P.P., Esposito V., Macia A., Sperano A., Meldelesi G.N., Picardia, June 2006. Severe amnesia following bilateral medial temporal lobe damage occurring on two distinct occasions. Neurological Sciences 27, 2, 129-33 doi, 10.1007 per second 10072-006-0614y. PMID 16816912, Squire and Schachter, 2002, ch1, Diana al., 2007, Matsumura al., 1999, Rolls and Xiang, 2006, Ekstrom al., 2003, Smith and Mizumori, 2006, O'Keefe and Nadal, Chu al., 2004, Morris al., 1982, Sutherland al., 1982, Sutherland al., 2001, Clark al., 2005, Solstad al., 2008, Maguire al., 1998, A.B. Maguire al., 2000, ABCDE Amaral and Lavinex, 2006, Cotter and Stephan, 1997, Moser and Moser, 1998, Winson, 1978, A.B.I. Chenbaum al., 2007, A.B. Bwatsaki, 2006, A.B. Bwatsaki al., 1990, A.B. Skaggs al., 2007, Lubinov and Siapis, 2009, Bwatsaki, 2002, Cantero al., 2003, Van der Wolf, 1969, Hutter and Lisman, 1993, Kahana al., 2001, Bwatsaki, 1986, Wilson and McNaughton, 1994, Jackson al., 2006, Sutherland and McNaughton, 2000, Bwatsaki, 1989, Ramon Y. Cajal, 1894, Hib, 1948, Bliss and Lomo, 1973, Cook and Bliss, 2006, Malanka and Bear, 2004, A.B. Nekazor al., 2004, Hampel al., 2008, A.B. Proul al., 2000, pages 105, A.B. Proul al., 2000, pages 107, Erickson al., 2011, Rosenzweig and Barnes, 2003, Burke and Barnes, 2006, Joseph, James A. Fisher, Derek R. Cheng, Vivian. Raimondo, Agnes M. Shikite Hale, Barbara, 2008. Cellular and Behavioral Effects of SCAB in Resveratrol Analogues, Implications for Reducing the Deleterious Effects of Aging. Journal of Agricultural and Food Chemistry 56, 22, 10544-51 doi, 10.1021/JF80227-9H. PMID 18954071, Joels, 2008, Fu al., 2010, Kahler, Schaefer M., Malta L.S., Dorfel D., Roleda N., Werner A. 2006. A meta-analysis of structural brain abnormalities in PTSD. Neurosi BioBu of Rev 30, 7, 1004-31 doi, 10.1016/Janibirov. 2006.03.004. PMID 16730374, Wright I.C., Rabe Hesketh, Woodruff P.W., David A.S., Maria M., Bullmore E.T. 2000. Meta-analysis of regional brain volumes in schizophrenia. Amj Psychiatry 157, 1, 16-25. PMID 10618008, Kempton M.J., Salvador Z., Runafo Mr., Geddes J.R., Simmons A., Franger S., Williams S.C. 2011. Structural Neuroimaging Studies in Major Depressive Disorder, Meta-analysis and comparison with bipolar disorder. Arch General Psychiatry 68, 7, 675-90 doi, 10.1001/Arch Psychiatry. 2011.60. PMID 21,727,252. See also MRI database at www.depressiondatabase.org, Campbell and McQueen, 2004, Garcia Segura, pages 170-71, Conrad C.D., 2008. Chronic stress-induced hippocampal vulnerability, the glucocorticoid vulnerability hypothesis. Reverend Neurossi. 19, 395-411 doi, 10.1515/Rev Neuro. 2008.19.6.395, Chang and Lauenstein, 2003, Slavta, 2005, Karuba al., 2009, A.B. Harrison, 2004, Goto and Grace, 2008, Boyer al., 2007, Lewis S., 1998. Etiology of transient global amnesia. The Lancet 352, 9125, 397-9 doi, 10.1016/s 0140-6736, 98, 01442-1. PMID 9717945, Chung C.P., Xu H.Y., Chao A.C., Chang F.C., Sheng Wei, Hu H.H., 2006. Detection of intracranial venous reflux in patients of transient global amnesia. Neurology 66, 12, 1873-77 doi, 10.1212/01WNL000219620.69618.9 d. PMID 16801653, West, 1990, Suzuki al., 2005, A.B. Jacobs, 2003, Jacobs al., 1990, Aboitis al., 2003, Rodriguez al., 2002, Colombo and Broadbent, 2000, Shetluworth, 2003, Nuenes, 1982, Portavella al., 2002, Vargas al., 2006, Brolio al., 2005, Mizunami al., 19